Gel electrophoresis. Gel electrophoresis can be used to separate DNA fragments. Electrophoresis uses an electric current to separate different-sized molecules in a porous, sponge-like matrix. Smaller molecules move more easily through the gel pores than larger molecules, while at Cold Spring Harbor Laboratory, Phil Sharp, Joe Sandbrook, and Bill Sugden developed the DNA electrophoresis technique using an agarose gel made from highly purified seaweed. This could be used to separate DNA molecules, ranging from several hundred nucleotides in length to over 10,000 nucleotides. The gel is submersed in a tank filled with a salt solution that conducts electricity. Using a pipette, DNA samples are loaded into slots made in the agarose gel. The DNA samples are colorless, but researchers add a blue tracking dye. This makes it easier to load the samples and visually track the DNA migration through the gel. The phosphate groups in the DNA backbone carry negatively charged oxygens, giving a DNA molecule an overall negative charge. In an electric current, the negatively charged DNA moves towards the positive pole of the electrophoresis chamber. The DNA molecules move through the gel by reptation, a reptile-like snaking action through the pores of the agarose matrix. Smaller DNA fragments migrate faster and further over a given period of time than do larger fragments. This is how DNA fragments can be separated by size in an agarose gel. Sharp, Sandbrook, and Sugden introduced the use of the fluorescent dye, ethidium bromide, to stain DNA. Ethidium bromide binds tightly to the DNA double helix and glows when illuminated with ultraviolet light. This lets researchers see where the separated DNA fragments end up. A photo of the gel can be taken for later analysis. The size of any DNA fragment can be determined by comparing it to markers, DNA fragments of known sizes. A map of restriction enzyme sites can be generated by cutting a piece of DNA with different combinations of restriction enzymes.